Epigenetics and disease. Epigenetics is the study of changes in gene function that occur without a change in the DNA sequence itself. One important epigenetic mechanism is DNA methylation. DNA methylation results from the attachment of a methyl group to a cytosine base within the DNA strand. This modification typically renders genes inactive, meaning they are turned off and not expressed. DNA methylation plays a crucial role in normal human development, helping guide cells into their proper functions. However, abnormal changes to DNA methylation are associated with several diseases, especially cancer, where incorrect gene silencing or activation can drive tumor growth and progression. Histones are specialized proteins that help compact the long strands of DNA so that they can fit inside the tiny space of the cell nucleus. Without histones, the human genome would be far too large to be properly organized within a cell. The combination of DNA wrapped around histones forms a complex called chromatin, which plays an essential role in regulating gene accessibility and expression. In addition to providing structural support, histones can undergo chemical modifications. These modifications can either upregulate, meaning increase, or downregulate, meaning decrease, the expression of nearby genes. This fine-tuning of gene activity through histone modification is absolutely critical for normal development and healthy cellular function. Embryonic stem cells are remarkable because they are totipotent, meaning they have the potential to give rise to all the different cell types of an entire organism. During development, specific genes are selectively expressed in different cells and tissues, allowing each part of the body to develop its unique structure and function. Meanwhile, other genes that are not needed for a particular cell type are epigenetically silenced to prevent their expression. However, there is a special category of genes known as housekeeping genes. These genes escape epigenetic silencing and remain constantly active because their functions, such as basic cellular maintenance, are necessary in all cell types. Genomic imprinting is a fascinating aspect of gene regulation where the expression of genes depends on which parent the gene was inherited from. In biallelic expression, both maternal and paternal copies of a gene are active and contribute to the offspring's traits. However, in monoallelic expression, only one copy of the gene is expressed, and the other is randomly inactivated. In some cells, the maternal copy may be turned off, while in other cells, the paternal copy is silenced. When a gene is imprinted, it means that the copy inherited through either the sperm or the egg is permanently inactivated and remains silent throughout the individual's life. This parent-specific silencing can have significant impacts on development and disease. Many of the genes that are subject to imprinting play a key role in regulating growth. This leads to an intriguing idea known as the genetic conflict hypothesis. According to this hypothesis, genes inherited from the mother and father may have opposing interests when it comes to the growth of the offspring. Imprinting of maternally inherited genes typically reduces the size of the offspring, which may serve to conserve the mother's resources. In contrast, imprinting of paternally inherited genes tends to increase offspring size, which may enhance the father's genetic success by producing larger, potentially more robust children. This tug-of-war between maternal and paternal influences is a central theme in the study of genomic imprinting. A striking example of genomic imprinting involves the deletion of about 4 million base pairs on the long arm of chromosome 15. Interestingly, the effects of this deletion depend entirely on whether it is inherited from the father or the mother. If the deletion is inherited from the father, the child may develop Prader-Willi syndrome. This condition is characterized by short stature, low muscle tone, small hands and feet, obesity, and intellectual disabilities. On the other hand, If the same deletion is inherited from the mother, the child may develop Angelman syndrome. These individuals often present with severe cognitive impairment, seizures, and an uncoordinated, ataxic gait. What's remarkable is that these two syndromes are indistinguishable at the level of DNA sequence. It is only the parent of origin that determines which syndrome emerges, highlighting the powerful influence of genomic imprinting. This image illustrates the contrasting physical appearances of two distinct syndromes caused by the same genetic deletion on chromosome 15. On the left, we see a child with Prader-Willi syndrome, which occurs when the deletion is inherited from the father. These individuals often have short stature, low muscle tone, and a tendency toward obesity due to an insatiable appetite. On the right is a child with Angelman syndrome, resulting from the same deletion inherited from the mother. This syndrome is marked by severe cognitive impairment, frequent smiling or laughter, and problems with balance and coordination. Although the underlying DNA sequence is identical, the parent of origin determines the outcome, an extraordinary example of how genomic imprinting can shape human development. This diagram visually explains how a small deletion on chromosome 15 leads to two very different disorders, Prader-Willi syndrome and Angelman syndrome, depending on which parent the chromosome is inherited from. On the left side, the deletion occurs on the chromosome inherited from the father. Because the maternal copy is normally silenced at the Prader-Willi region, the loss of the paternal copy results in no functional gene expression from that region, leading to Prader-Willi syndrome. On the right side, the same deletion occurs on the maternal chromosome. Since the paternal copy is normally inactive at the Angelman region, this leaves the individual without an active copy, leading to Angelman syndrome. This example highlights how imprinting controls which gene copies are active or inactive and illustrates why parent of origin matters so significantly in genetic expression. Beckwith-Wiedemann syndrome is a growth disorder linked to errors in genomic imprinting. In 20 to 30% of cases, it results from the inheritance of two copies of a chromosome from the father and none from the mother, a situation known as paternal uniparental disomy. This genetic imbalance leads to the overexpression of the insulin-like growth factor 2 gene, or IGF-2, a powerful driver of growth. The manifestations of Beckwith-Wiedemann syndrome are evident at birth. Affected newborns may be unusually large for their gestational age and may also experience low blood sugar, an enlarged tongue, creases on the earlobes, and various intestinal birth defects. This condition clearly demonstrates how improper genomic imprinting can disrupt early development. Russell-Silver syndrome is another disorder linked to abnormal genomic imprinting. About 1/3 of all cases are associated with imprinting abnormalities on chromosome 11p15, 5, and around 10% are caused by maternal uniparental disomy, where both copies of a chromosome are inherited from the mother. A key feature of this condition is the downregulation of insulin-like growth factor 2, or IGF-2, which plays a central role in fetal growth. As a result, children with Russell-Silver syndrome often present with growth retardation, proportionate short stature, and a noticeable difference in leg length. They may also have a distinct small, triangular-shaped face. This condition further illustrates how precise regulation of gene expression by imprinting is essential for normal development. There is growing evidence that nutrition can influence epigenetic changes, and these changes may affect not just one generation, but potentially multiple generations. When nutritional deprivation occurs, especially during critical developmental periods, it can leave long-lasting molecular marks on the genome. Although the exact mechanisms are still not fully understood, one possible target of these modifications is the IGF-2 gene, which is involved in growth regulation. Studies suggest that in utero exposure to chemicals or malnutrition can trigger similar epigenetic modifications, which may increase the risk of disease later in life. This highlights the profound impact early environmental conditions can have on lifelong health through epigenetic pathways. Early life experiences, particularly those involving maternal care and environmental stress, can have a lasting impact on gene expression through epigenetic mechanisms. Alterations in DNA methylation may help explain why stress during critical periods of development can influence behavior well into adulthood. One notable example is fetal alcohol syndrome. When a fetus is exposed to alcohol in the womb, it can disrupt normal skeletal and neural development. Ethanol exposure during this time may also interfere with the expression of enzymes called DNA methyltransferase, which are essential for maintaining proper methylation patterns. These disruptions can contribute to a range of developmental and behavioral problems later in life. One of the most well-known examples of an epigenetic abnormality tied to genetic disease is found in Fragile X syndrome, which is linked to the FMR1 gene. In typical individuals, this gene contains approximately 35 repeats of a specific DNA sequence made up of cytosine, guanine, and guanine, abbreviated as CGG. However, when the number of CGG repeats increases significantly, it can trigger disease. This happens through a process called methylation-based silencing. Interestingly, the acquisition of this silencing is stochastic, meaning it occurs by chance. While having more repeats increases the likelihood that the gene will be silenced, it does not guarantee that silencing will occur. This randomness makes diagnosis and prediction more complex, underscoring how epigenetic processes can add another layer of variability to inherited conditions. This diagram shows how Fragile X syndrome develops through both genetic mutation and epigenetic silencing. Under normal conditions, the FMR1 gene produces messenger RNA, or mRNA, which is necessary for healthy brain function. However, in Fragile X syndrome, the FMR1 gene undergoes a mutational expansion, an increase in the number of CGG repeats in its sequence. This expanded region becomes densely methylated, meaning that multiple methyl groups are attached along the DNA strand. As a result, the gene becomes silenced, and no mRNA is produced from FMR1. This loss of gene expression disrupts neural development and leads to the cognitive and behavioral symptoms associated with Fragile X syndrome. It's a clear example of how a genetic mutation can trigger an epigenetic response with profound biological consequences. Facioscapulohumeral muscular dystrophy, or FSHMD, is a genetic disorder that highlights the role of epigenetics in muscle disease. This condition arises from a loss of normal DNA methylation. Specifically, it involves the deletion of a nucleotide repeat sequence in the DUX4 gene. Under normal conditions, methylation helps silence this gene. But when the repeat is deleted and methylation is lost, DUX4 becomes inappropriately activated. The resulting gene expression disrupts muscle function and leads to the progressive muscle weakness that characterizes this form of muscular dystrophy. FSHMD offers a powerful example of how the absence of epigenetic regulation, not just genetic mutation, can cause disease. This diagram shows how facioscapulohumeral muscular dystrophy, or FSHD, results from a combination of genetic mutation and epigenetic disruption. In a healthy individual, the DUX4 gene is located in a region that is densely methylated, preventing it from being expressed. However, in FSHD, there is a mutational contraction of the repeated DNA region near DUX4. This contraction leads to a loss of methylation, allowing the DUX4 gene to become transcriptionally active. As a result, mRNA is produced from the DUX4 gene, which should normally remain silent. This abnormal activation causes muscle degeneration, leading to the progressive muscle weakness seen in individuals with FSHD. It's another clear demonstration of how epigenetic control is essential for protecting tissue health and function. Twin studies offer a unique window into the world of epigenetics. Because identical twins share the exact same genetic sequence, any differences that arise between them over time are likely due to environmental influences and epigenetic modifications. As twins age, scientists have observed increasingly significant differences in their somatic cells, particularly in DNA methylation patterns. These differences become even more pronounced when twins live different lifestyles, such as eating different diets, managing different stress levels, or being exposed to different environmental toxins. These findings suggest that epigenetic changes are not only dynamic and responsive to the environment, but they also play an important role in the aging process. To study epigenetic diseases, scientists rely on molecular tools that detect chemical modifications on DNA and histone proteins. These modifications encode important regulatory information, much like bookmarks in a genetic instruction manual. One common technique is bisulfate conversion, which is performed before DNA sequencing. This process helps identify which cytosine bases have been methylated. Another important method involves using antibodies to detect specific histone modifications. These antibodies can recognize chemical tags on histones, revealing how genes are being turned on or off in different cells. Together, these molecular approaches provide valuable insights into how epigenetic changes contribute to health and disease. Epigenetic abnormalities are a hallmark of many cancers. In tumor cells, DNA methylation patterns are often disrupted in ways that promote unchecked growth. One such abnormality is hypomethylation, where genes that drive cell division become overly active. These genes, when not properly silenced, can lead to excessive cell proliferation and tumor formation. On the other hand, tumor suppressor genes, which normally act as brakes on cell division, can become hypermethylated. This excessive methylation silences their protective functions. Key examples include the RB1 gene associated with retinoblastoma, the BRCA1 gene linked to some inherited breast cancers, and the MLH1 gene often involved in colon cancer. These epigenetic changes can be just as important as mutations in driving cancer development. Epigenetic screening is emerging as a powerful tool in the early detection of cancer. Because changes in DNA methylation and histone modification can occur long before a tumor becomes clinically detectable, identifying these epigenetic alterations may allow for earlier diagnosis. Detecting cancer in its early stages greatly increases the chances of successful treatment. As research continues to uncover reliable epigenetic markers of cancer, these screening tools could become a routine part of preventative healthcare, offering a more sensitive and specific method for spotting cancer at its onset. One of the most exciting developments in epigenetics is the possibility of reversing harmful gene expression patterns with targeted treatments. Pharmaceutical agents are being developed to counteract the epigenetic changes that drive disease. For instance, DNA demethylating agents such as 5-azacitidine can remove methyl groups from DNA, potentially reactivating silenced tumor suppressor genes. Histone deacetylase inhibitors represent another class of drugs that help restore proper gene expression by loosening tightly wound chromatin and making genes more accessible for transcription. In addition, researchers are exploring the use of microRNA or mRNA coding to further regulate gene expression at the post-transcriptional level. These therapeutic strategies offer real hope for treating cancers and other diseases rooted in epigenetic dysfunction. One of the most promising aspects of epigenetics is that, unlike mutations in DNA, epigenetic modifications are potentially reversible. This means that certain diseases driven by these changes might be treated, not just managed, through targeted therapies. Drugs that reverse harmful epigenetic alterations are already in clinical use. DNA demethylating agents work by removing methyl groups that silence important genes, while histone deacetylase inhibitors help reopen tightly packed chromatin, allowing the essential genes to be expressed again. These treatments offer new hope for patients with cancers and other conditions rooted in epigenetic disruption.